differences because as we've talked about previously, the chromosomes in the DNA in eukaryotes are a lot more complex and there is a lot more DNA, and so there's got to be a lot more regulation involved. So as we talked about in last chapter, bacteria utilize repressor proteins as a way to prevent RNA polymerase from transcribing a gene, and activator proteins help to start or turn up a gene, right? However, due to the large size and increased complexity in eukaryotes, they use a different mechanism for regulating this transcription. Eukaryotic DNA is packaged into nucleosomes, forming chromatin. And so these nucleosomes are wound DNA that kind of prevent and block the ability of RNA polymerase to reach the promoter. When the gene needs to be turned on and transcription needs to take place, chromatin structure needs to be changed to allow access to that promoter. Thus, regulatory proteins function by changing the chromatin structure and allowing access to the gene. So this blocks or removes the blocks that are in the way of transcription. So as we talked about in previous slides, there are regions upstream of the gene that are involved in either recruiting the transcription machinery itself or in regulation through repressing or enhancing gene transcription. And so how scientists sort of figured this out is throughout the upstream region of the gene, they introduced point mutations at every single nucleotide, well, almost every single nucleotide. The little black dots here are ones that were not tested. However, the majority of these nucleotides upstream of the gene itself, they introduced point mutations to change the sequence at just a single base pair. And as you can see in this slide, for the most part, it had no effect on transcription. So changing an A to a G at this nucleotide, for example, didn't really cause the gene to be transcribed any more or any less. However, there are three regions that they identified upstream of the gene itself that when you introduced a single nucleotide change to one of the base pairs, it caused a reduction in the expression of that gene. So these two regions up here are called the proximal elements, promoter-proximal elements to be specific. So that's this green region and this gold region, as well as this red region, which is the Tatabox, which we had talked about earlier is what helps to recruit and kind of set up the start line for transcription in eukaryotes. So let's look at a pathway, an example here of how regulation of these genes in eukaryotes takes place. In yeast, the galactose pathway, or GAL for short here, is a way for yeast to convert sugar into usable energy. This is very similar to the lactose pathway that we discussed last chapter. However, instead of using lactose, it uses galactose, which is another complex sugar. There are 4 genes in yeast that work to convert galactose into usable energy via a process called glycolysis. These are GAL1, GAL2, GAL7, and GAL10. Just like the lactose pathway, if there's no galactose around, these genes are not transcribed. So there's no point to have them if there's nothing to convert. In conditions where there is high galactose and low glucose, which is the easily convertible sugar we talked about before, these genes are activated and actually go to work. GAL2 brings galactose from the exterior of the cell into the interior of the cell. GAL1, GAL7, GAL10, and then GAL7 again, then process the sugar to form glucose 1-phosphate, which can then be converted into energy via galactose metabolism. However, there's a gene that is missing from this pathway that is also important for this regulatory mechanism in the galactose pathway. That's the GAL4 gene, which we'll talk about next here. So when galactose is present in a cell, the transcription of the GAL genes is 1,000-fold higher than when there's not galactose present. However, as we see here in yeast, a mutation in the GAL4 gene causes no difference in the expression of the GAL genes, whether or not glucose is present or not. So each of the GAL genes, that was GAL7, 10, 1, and 2, or 1, 2, 7, and 10 in order, have 2 or more GAL binding locations upstream of the gene. So that's here for GAL7, here for gal10, here for gal1, and then here for gal2. Each of these binding locations is a 17 base pair long sequence, and it binds a single gal4 protein. If these sites are mutated or removed, then transcription doesn't happen. These sites are called upstream activator sequences, or UAS. So this gal4 protein has two domains. One that is responsible for binding DNA, and one that is responsible for activation of transcription. So we see the binding domain here and the transcription activator domain up here. By removing the activator domain, we see that binding of the actual DNA binding domain by itself results in no transcription of the LAC-Z operon here, which is our example. However, there's a second regulatory protein similar to the gal4 protein, and it's called LEX-A. When LEX-A binds to a DNA binding domain, we similarly see that there's no transcription, indicating that these domains their only purpose is to bind to the DNA. However, if we create this hybrid where we take the LEX-A binding domain into the activator site from the gal4 gene and kind of slap them together here and make this hybrid activator protein, that LEX-A binding site is able to bind to the DNA, and this activator from a different protein, from the gal4 activator protein, is enough to turn on activation of the LAC-Z gene. So this suggests that these activator proteins are kind of ubiquitous, and they're able to work on multiple different genes. They're not specific like a transcription factor to one gene. So we know that gal4, its job is to activate transcription of this gal operon, which in turn turns galactose into usable energy. So what controls when gal4 is active or inactive in enhancing or allowing activation of this gal operon? So looking at yeast, scientists identified another protein called gal80. And what gal80 does is when there's no galactose present, it binds to the active site here of, let me get my pointer, active site here of gal4, preventing it from allowing activation of the gal operon and then subsequent metabolism of galactose. However, there's got to be a way to turn this mechanism off, right? And so in the presence of galactose, another gal regulatory gene called gal3 has an allosteric site, like we talked about last chapter, where it senses whether or not galactose is present and changes its conformation. So when galactose is present, that gal3 gene can then release gal80 from the active site of gal4, allowing for it to act as an enhancer or act as an activator of this gal1 operon and then subsequent metabolism of galactose to proceed. So now that we know how gal4 is regulated, how does it aid in transcription? gal4 is responsible for recruiting transcription machinery to the start site. As you can see in this diagram, the UAS, or the upstream region where gal4 binds, is quite a ways away from the promoter itself. So the DNA in this example folds over on itself to bring gal4, this activator, closer to the TATA box, or the place where transcription starts. What gal4 then does is it helps to recruit these proteins and fit them together and place them in the TATA box or the promoter region, allowing for transcription to proceed. gal4 itself does not have any job or any role in actual transcription, just in recruitment of the complex, the RNA polymerase II haloenzyme, to the proper site to allow for transcription to proceed. So let's talk about one of the major differences between prokaryotes and eukaryotes in gene regulation. And this is the fact that prokaryotes are single-celled organisms. They're very simple. They don't have, multiple chromosomes, a single circular chromosome. However, eukaryotes are much more complex. And so if we look at a complex organism such as a human, there are many different tissues that these different genes are going to be needed or not needed for. So the same genes that are required for the formation and proper cellular function of brain tissue likely aren't going to use all the exact same genes as in different tissue, such as heart tissue. So how do you turn on and turn off these genes in different cell types based on whether or not you need them or not? So as a simplified example, we're going to look at yeast. So yeast has three different types of cells, or three different yeasts. You have an A type of yeast, which is indicated as A here. You have an alpha type, which is indicated here, and then you also have an alpha A hybrid, or alpha A yeast type, which we'll get to in a second. And so the A1 type and alpha 1 type, so both A and the alpha types, are haploid. So they only have one copy of each chromosome. That A alpha that I mentioned a second ago, that is diploid and it has two copies of each chromosome. And so the difference in these cell types for yeast is their ability to mate. So an A cell type for yeast is able to mate and only with alpha pairs. So A always looks for alpha to mate with, and then the same is true for alpha. Alpha only looks for A to mate with. Each of these two yeast cell types excrete pheromones that aid in reproduction with their prospective mate. So alpha will release a pheromone that stops the cell cycle in A yeast cells. And so that enables it to mate with it without that yeast cell still going through its cell cycle process and kind of interrupting mating. And the same is true in the opposite way, where A release pheromones that stop the cell cycle for alpha and allows them to mate with their respective pair. The A alpha hybrid that we had talked about earlier, diploid, that does not mate at all, and it's not responsive to either of these pheromones produced by the A cell type or the alpha cell type. So this cell type and this whole mechanism is controlled by a single locus called the MAT locus, or MAT locus. So analysis of mutants of these yeast strains has revealed that successful mating relies on some genes that are not part of the MAT locus. As we saw in previous examples, there are genes located upstream or distantly on chromosomes that often play a part in regulation of these operons, or these collections of genes that are required in these pathways. So one set of genes is expressed only in the A individuals, and then a separate set of genes is only expressed in the alpha individuals. The MAT-A allele expresses only a MAT-A protein, while in the alpha cells, there are two genes in the MAT-A alpha that are expressed only in those cells, and that's the alpha 1 and the alpha 2 genes. So let's look at all three of these cell types and how regulation of cell-specific genes takes place. The simplest case, as shown here on this slide, is the A cells in the MAT-A1 locus. And so in these A cells, a single protein is transcribed called A1. And in the A cell itself, A1 doesn't really have a function. It just is kind of hanging out. And MCM1, which is an activator of transcription, will bind naturally to the A-specific gene regulatory region, and it will allow or activate transcription of those A-specific genes. Now, if we look at the alpha cells, they want to transcribe only the alpha-specific genes and not the A-specific genes. So what it does is at the MAT-A locus, alpha 2 and alpha 1 proteins are transcribed, and what alpha 2 does is it binds to MCM1 and it prevents it from transcribing the A genes, because remember, it only wants alpha genes. Instead, A1 will also bind to the MCM1, to different copies or different proteins of that same type, and it will recruit it to the regulatory region of the alpha-specific genes. And that will allow for transcription of only the alpha genes and repression of the A-specific genes. In both these cell types, haploid-specific genes are always transcribed because these cells are haploid. Now, what happens in these A-alpha hybrids, or these diploid cells that have both the MAT locus for A and the MAT locus for alpha? So remember, in the MAT locus for A, there was this A1 protein that was made and really didn't have a role in haploid cells. However, in this hybrid, this diploid, A1 actually has a role. So A1 will interact with A2, and they will bind upstream of those haploid cells that are required for the haploid life cycle, and they will prevent the transcription of all those haploid-specific genes. In addition, the alpha cells will bind to MCM1, and that will prevent any transcription of these A-specific genes that we saw in the MAT-A cell type. And since there's no alpha-1 protein being expressed in these haploid cells, that doesn't allow for the alpha-specific genes being expressed. And remember, these A-alpha hybrids, they're not mating anymore because they've already mated once. And so they don't want either of the A or the alpha genes to be expressed. They just want to go through their life without having to worry to mate. So this complement of the genes from the A type and from the alpha type prevents any sort of transcription of genes required for further mating. So one of the methods, or one of the ways that eukaryotes and prokaryotes different, as alluded to earlier, is how the DNA is packaged. And so in prokaryotes, the DNA is very simple. It's the small circular chromosomes. There's no need to really package it into any complex structures, and it allows easy access for the transcription machinery to get there. And that way, these regulatory proteins that we talked about in the last chapter are all that's really needed to turn genes on and off, because there's not a lot of regulation needed. However, this is very different in eukaryotes. In eukaryotes, the DNA is bound up into chromatin, and chromatin is made up of smaller components called nucleosomes. And these nucleosomes are generally about a 150 base pair sequence of DNA that's wrapped around a protein complex of these histones, which are kind of a octamer of protein subunits. And they're really, their main role is to just kind of package the chromosomal DNA. When this DNA is packaged, there is really limited access for the transcription machinery to get in there. So if you kind of look at these simple drawings and even these more complex chromatin formations here, you see that the DNA is wrapped around tightly around these nucleosomes, and for RNA polymerase and the transcription machinery to get in there, it's very limited in the access that it has to activate transcription. So this acts as another way of preventing transcription of genes that don't need to be transcribed at that time. What we're finding now that's really interesting is that these histone modifications, or the way that this DNA is packaged, and some of the way that the proteins involved in the structure of DNA, this is an inheritable trait. And so parents will have certain formations of histones and their nucleosomes that is passed down to their children. So genes that are not transcribed because of the way the DNA is packaged in a parent, that trait can be passed down to their children, and is another way that traits are inherited or passed down from mother to child. And so on top of DNA being the inheritable material, we're seeing that these epigenetic or outside genetic traits are also passed down from mother to child, or from father to child. So we see that histones and the nucleosome are involved in regulating access to DNA by wrapping, the DNA wraps around these protein complexes. So how is this kind of access granted or not granted to a gene? So there's a process called chromatin remodeling, in which proteins act on the DNA nucleosome complex and kind of slide the protein further down the sequence to allow genes to be transcribed. So if you see in this case here, we have a gene that we're interested in transcribing. It needs to be transcribed for response to some external stimuli, and there'll be some sort of proteins that we'll get into in a little bit here that act on this nucleosome and kind of allow the whole DNA to wind around and slide past the histone and allow access for the transcription machinery to sneak in here and start transcribing that gene. So how does this mechanism work? I had said that there's proteins that kind of act upon these histones and allow it to slide back and forth and allow access to genes or not. Well, looking at these histones more closely, there are actually these tails that protrude from them. And at the end of these tails, there are amino acids that can be modified by adding acetyl groups, a process which we call acetylation. And in this figure here, we see the acetyl groups labeled as an A. And so acetyl groups and acetylation work in both ways. So acetyl groups can be added to these tails of the histones, or they can be removed from these tails of the histones, depending on whether or not this gene needs to be transcribed or does not need to be transcribed and needs to be regulated. So looking at the modification of these histones via acetylation, we see that there's actually a direct correlation between the amount of acetylation that takes place on these tails of these histones and the amount of transcription that takes place. And so it turns out that adding a whole bunch of acetyl groups to these histones kind of unbinds the nucleosome and spreads out the DNA and allows that unwinding process to take place. So as you add more, there's more space for the transcription machinery to get in here and start transcribing genes that were previously inaccessible because of the way that the nucleosome was formed. So as I just stated in the last slide, acetylation is reversible too. So you can add acetyl groups or you can remove acetyl groups depending if you need to turn on genes or turn off genes. So we'll look at this with the Gal4 operon that we talked about as an earlier example. So with this Gal4 operon that we talked about before, when glucose is present, the MIG-1 protein binds to the repressor site to ensure that the galactose pathway isn't transcribed. And there's enough sugar available as glucose to not need transcription of the galactose genes. So the MIG-1 protein also recruits another protein called TUP1. And what TUP1 does is it targets and it removes acetyl groups from nearby histones or nucleosomes. And what this does is, remember we had said earlier that adding acetyl groups allows those access to transcription or genes to be transcribed because it kind of spreads out the histones or the nucleosomes. But removing these acetyl groups allows those histones and the nucleosomes to come closer together and prevent further access of that gene to the transcription machinery, which would be required for gene transcription. So this kind of shuts down the Gal operon in a second way in addition to these repressor proteins that bind. As is stated earlier, histone state is inherited by the daughter cells as well. During DNA replication, the histone complex is split, and the old modified histones are passed down to each resulting daughter cell. As you can see, the purple here is the old histone, and it's passed to each forming daughter strand. These passed down histones guide the modification of the newly synthesized histones that are on the newly synthesized strand and help to determine the pattern of histone modification and gene access that was exhibited in the parent. So another form of modification of access to genes, or epigenetic modification, we should say, is methylation. And so methylation occurs at many GC pairs within the genome, and it only happens at GC pairs. So semi-conservative replication results in daughter strands that are methylated on one of the two strands, right? So this strand is, even though both strands here are methylated, one strand goes to this daughter strand and has only one methylation spot, and this strand goes to this daughter strand that only has one methylation spot. However, there's an enzyme called DNA methyltransferase that reads the unmethylated strands, and it kind of pairs up these methylation points and will then go on to methylate the strand to match what they were in the parent cell. And so methylation generally acts as a way to turning off genes. As you can see, if you have this big methyl group that's sticking out of your DNA strand, then when the transcription machinery comes and tries to either bind here to activate transcription, or it starts reading down and reaches these methyl groups, it's going to inhibit the transcription machinery to proceed. We talked briefly in the previous chapters about the proteins that help to start transcription. There's a class of these proteins called Enhanceosomes, and these proteins act synergistically to activate transcription of a gene. These proteins act to help create the proper form and shape of transcription machinery to bend the DNA around the transcription machinery. The form of the transcription machinery is key to high levels of transcription, and thus these enhanceosomes, which are indicated as these DNA bending proteins here, are very key to high levels of transcription of genes. So let's look at an example of these enhanceosomes using the beta interferon gene, which is normally inactive, but is activated upon viral infection. So it's a viral response gene. So upon this viral infection, the enhanceosome recruits the transcription machinery upstream of the gene of interest. So this enhanceosome in complex, transcription complex, recruits another gene called GCN5. What GCN5 does is it temporarily binds to this bound transcription complex, and it acetylates the gene or the nucleosomes or the histones that are nearby the gene. Which, remember, adding acetyl groups allows this histone to kind of unwind and move away and allow access to the gene that we're interested in transcribing. After acetylating the nearby histones, GCN5 leaves and allows CBP, which is the co-activator of this complex, and SWI-SNF, which we'll refer to as SWI/SNF, and RNA polymerase II to combine and add to the transcription start site here. SWI/SNF further moves this histone away from the start of the transcription site, which allows for the final ingredient, the TBT, or the TATA binding protein, to bind to the TATA box, which is now exposed because of SWI/SNF, and proceed to transcribe the gene. As we talked about earlier, enhancers bind to regions of DNA and enhance transcription, but they also, as we saw in the example where we took the DNA binding domain and the activator site and recombine them, they're not very specific, and they act over a very large region of DNA. And so how do you prevent an enhancer from transcribing a nearby gene if you don't want it to, if it's not supposed to be enhancing, say, gene from promoter 1, but only gene from promoter 2? To prevent this non-specific enhancement that we are talking about or trying to avoid, enhancer blocking insulators, which we denote here as this kind of pink egg, have evolved. These proteins bind between the enhancer and the gene that you don't want enhanced, allowing only the enhancer to work on the gene that you do want to be transcribed or the enhancer activity to take place on. So how does this work? The current model of how these enhancer blocking insulators work is that they cause the formation of these DNA forming loops. The enhancer binds within a loop, and it's only able to enhance transcription on genes found within the same loop as it. It doesn't have this ability or transacting ability to enhance a gene that is found in a different loop. So we can see that when these enhancer blocking insulators form, they form and they bind to multiple enhancer blocking sites on different parts of the DNA and kind of confine enhancement only to localized regions as opposed to long stretches of DNA, which enhancers are naturally capable of doing. So let's delve a little further into that yeast mat example from earlier and look at the activation of certain genes. So yeast haploid cells are able to switch their mating type from A to alpha and vice versa, and this can happen as often as every cell cycle. This was very important for these yeast cells because, remember, mating can only happen between A and alpha. So if you're an A cell type and there's no alpha around, then you're forever prohibited from mating. So being able to switch cell types in order to mate with nearby other or opposing cell types would be very beneficial. So how does this happen? Well, the active MAT-A locus is what is used to create either the genes necessary for the A cell type, or this could also be the MAT-alpha active locus, which would make the alpha gene or the alpha cell type. However, both of these A and alpha cell types also have the genes for the alpha and the A cell type that are silenced. So they're not expressed, but they're there and they're available. So if the yeast wants to produce both offspring, say it's produced quite a few offspring of the A type, but realizes that there's a need for these alpha cells to also produce, what can happen is the DNA is replicated for this MAT or the HML alpha locus. So this is that silenced set of genes that contains everything needed for an alpha set but isn't being normally expressed. And what it will do is it will recombine with the original MAT locus and move this MAT locus out and insert this MAT A locus, resulting in the subsequent offspring of the cell being the alpha mating type, because it replaced the original cells with these MAT A genes. Now I think this is an instance where the slides are mislabeled. I believe this is supposed to be MAT alpha, and this just shows that that can also happen in the other direction. So pretend this was alpha instead of A, but the HMRA genes is a copy of the A-specific tissue type or A-specific yeast cells, and that also is able to be copied and replace the alpha, pretend this was labeled right, and produce A mating types as opposed to the original alpha mating types. So in either case, they can switch back and forth between the A yeast cells or the alpha yeast cells. Now we're going to talk about an interesting phenomenon called genomic imprinting. Parental imprinting or genomic imprinting is selective turning off or on of genes based on if you inherited them from your mother or from your father. The way it works is that during development of a gamete, methyl groups are added to the DNA of genes in one sex only. This addition of a methyl group shuts down the ability for the transcription machinery to bind and prevents the genes from being expressed, and this can be a long-term prevention of expression. Let's look at an example of this phenomenon by the LGF2 and H19 genes in mice. In mice, LFG2 is expressed only if it is inherited from its father, and H19 is transcribed only if it's inherited by its mother. Methylation of the ICR locus prevents the CTFC protein from binding. This prevention of binding to the ICR site allows the enhancer to work on the IGF2 gene, but it doesn't allow transcription machinery to get in and transcribe the H19 gene. Conversely, if the ICR site is not methylated, the CTCF protein binds to the ICR site and acts as an enhancer blocking insulator by forming those DNA loops that we had talked about earlier. So this prevents this enhancer from working on a piece of DNA that was in an opposite loop, because remember, these form loops and only allow the enhancer to work in the same loop. And since this methylation hasn't happened upstream of the H19 site, the actual transcription machinery can still sneak in here and transcribe the H19 site. So this weird mechanism of parental imprinting explains how some situations where mutations do not affect the offspring's phenotype. A mutation in an imprinted gene that has been inactivated shows no change in the phenotype. And this makes sense, right? Because the gene was never transcribed anyway, so a mutation there isn't going to change any proteins or associated phenotypes of the offspring. However, if you have a non-imprinted gene that isn't silenced by this mechanism, so here they still denote it AA, which is a little confusing, but you can see that there is no imprinting or blocking of or expression of only one gene, then you do have an affected outcome, because both of those copies of the gene are expressed, and so you do get those mutated proteins or a change in the phenotype of the offspring. So that concludes Chapter 12. We will be going over Chapter 12 questions that you may have, since you weren't able to really stop me and ask for clarifications. So I will bring this PowerPoint and we can address any figures that you didn't think that I explained fully or have questions about, and we'll also review on Tuesday. So this will be your last chance to ask any questions in class before the final exam next Thursday. But remember, you can always schedule office hours to come visit me or come to my posted office hours right after class.